Hello there, immunology. Let's get started. So, this is chapter 5, only the good parts. Yeah, of course. Yeah, right? What else am I going to show you? Right. So, these are the genes that come together to make the different chains. And there was one mistake in the version that was posted before, but I've corrected it here. Let's just actually spell that out. Beta. There we go. It had a little alpha there, and that was not right. So the T cell receptor beta chain is like the heavy chain, because just think big beta, big old beta. Yeah, and that's fine. So it's a heavy chain, and the giant difference is, wait for this, because this is going to be amazing. Variable genes, or variable exons rather. So we're talking about the exons that are going to come together and that will then form the gene. We'll then make RNA and we'll make protein, like genes work. The variable is about 95 amino acids worth of sequence, plus the little bit of 5' UTR. There's sometimes a leader on there as well that doesn't get translated, but it's like the untranslated region of the messenger RNA. But the actual coding bits, which are the important bit that we care about, are the variable for 95. The diversity segment is D. And diversity is worth all of about 5 amino acids. So, yeah, count them. It's the D chain that is different. So D is the difference. Yeah. Yeah. And, yeah, why not? D is the difference. That makes a heavy chain different from a light chain. There's also a different sort of constant region on there. There's a heavy chain version of a constant region, which tends to be the actual larger part. But this is the one bit that's actually extra. So there you go. Now you know. So variable, diversity, and then the J is the join region. Yeah, that's pretty small, about 10 to 15 amino acids. And together, these things form the antigen binding site. So the variable domain, if you like, of the antibody or of the T cell receptor. So, like an antibody, remember, T cell receptors have a heavy chain and a light chain. So it's big beta is the heavy chain, and little alpha is the light chain. Fine. And with antibodies, we just call them heavy and light. Yeah. Yeah, I don't know what else to say about that. That's pretty good. Yeah, solid. Yeah. All right. And so these things are going to come together by a process called somatic DNA recombination, which is literally DNA recombination. We're going to take out regions between some of these. So I'll show you. It's coming up in a couple of slides. We're going to look at the pictures because the pictures are so nice. So, refresher. This is what one of those variable domains looks like. And in the variable domain, there are those 3 hypervariable regions, 1, 2, and 3. As you can see here in red, and you can see they make little loops that then fold up and end up right next to each other. And that's the bit that usually makes contact with the antigen. Not always. There are so many weird antibodies out there that'll, bind the side of the antibody will bind to something, and that's fine. The antibody still works, even if it's not the bit that's supposed to bind that actually binds. Yeah, because an antibody's job is to stick into something. You'll see people use a fork. Some people will stab something with it. Some people will do this thing like with vegetables where they press the fork down on it, and so it just like mushes into the vegetables. Whatever, that works. It still picks stuff up. Yeah. So same deal with antibodies. They can pick things up in different ways, and doesn't really matter. Just a question of style, and you get the same, same nutrition from the meal. All right, so where does this map out onto those exons that are going to come together to make our finished antibody light chain or heavy chain gene? Well, the 3 hypervariable regions could come from the variable gene segment, so the variable exon, the V exon. But the third one can either come from this, or the third one can come from the join segment. And either way, whatever, it's fine. Yeah. So there are two possibilities that can give you the hypervariable region, and it's down to where the cell decides to randomly splice. And it'll probably splice half of them, that have this bit, and half of them that have this bit. It's going to be one or the other. You don't get both hypervariable domains, as far as I'm aware. Yeah, that's just how it works. All right. So here we go. So to make a light chain, there's no diversity in light chains, no diversity region. Yeah, so it misses out that whole 5 amino acids. So it makes them light. Yeah, light and nimble. Yeah. But you can see also that the constant region or constant exon is relatively small in a light chain and relatively large on a heavy chain. That's the actual difference. But here's our other one. So the D and the J are here, and this is the Wikipedia page showing the same thing. And so this is actually showing the order in which these things happen. So think of making this recombination like making a party. How does a party start? Yeah, well, it starts when Kesha walks in, for one thing. You're not old enough to even get that. But the other thing is that it starts when the DJ starts. So you got D over here, you got J over here. Got to bring those together. So we're going to take the diversity and join regions together, and that is the first recombination that happens in the process of building an antibody or a T-cell receptor. So you work on the heavy chain first. Got to take care of the heavy stuff before you get into that light stuff. And the first step is DJ. Yeah, D and J are going to join. Great. Next step is that we're going to take this DJ, or we're just going to take the J if we're a light chain and we don't have a DJ. Think, yeah, like, I don't know, light jazz or something like that. No DJ required. I kind of like light jazz, but whatever. Yeah. And so the next thing is we're going to bring whatever is attached to the J and the J together, and we're actually going to splice those together. And by splicing together, I mean we're literally going to cut out all the intervening DNA between that random variable domain and that random join region, or join and diversity region, and we're just going to put those two together, throw away the rest of the DNA, and it's going to be gone. Once that recombination happens, the cell cannot go back. And it's not chopping out nothing either. It is chopping out many, many other D and J and V regions. It just selects 3 at random. It's going to put them together, and better hope for the best, because you can't go back. Yeah. The fortunate thing is that the cells that are doing this do not last long. So if they make a loser of a combination, it's no big deal, because they're going to die soon. So hooray. Yeah. Yeah, immunology fills you with hope. Yeah. And so DJ first. DJ first. I mean, come on. Yeah. Yeah. You've been to a party. Yeah. Sometime before COVID, anyway. Then you get your J and your V together, or your V and your D and J together. And then the next step is going to be you're going to stick that constant region on. And so these are all just exons, remember. And so we're going to put the exons on, and then bam. You got yourself either a light chain or a heavy chain. So same process that you're seeing here. This is antibodies. Same process for T-cell receptors. The alpha chain works like a light chain. It's got V, J, and C. The big bad beta chain has got a diversity region in there, and it's a little heavier. It's the equivalent of a heavy chain. That's it. That's the idea. So, yeah. Makes sense? Hopefully. Yeah. I don't know. It's not hard. It's just like a thing to remember. All right, and light chains, there are kappa versions and there are lambda versions, and each B cell only gets to make one. It's going to pick a kappa or it's going to pick a lambda, and it's going to shut down. If it picks a lambda 27, it's going to shut down all the other lambdas, and it's going to shut down all the kappas, because there can be only one. Highlander rules apply to antibodies. Yes, fine. You're also too young to get that one. Oh my gosh, yeah. All these are missing today. Wow. All right. So, how many chains do you get? Here is the answer. So, of the kappa light chains, 34 to 38. You're not going to need to remember these, but this is like, you could do a little math here. So think how many combinations you can make. So you can make 30, let's say 38. And the difference here is variation in different people. You may be born with 35 light chains, and somebody sitting next to you might have been born with 36. Yeah. They can just check their privilege, and they may be able to make an antibody type that you just are genetically incapable of making. And that's, again, that's genetics. You get what you get, and you got to make the most of it, right? Yeah. Yeah, any of those kappas can combine with any of the join regions, and any of those can combine with a constant region. But for kappa chains, there's only one constant region, so that's simple at least. So if you wanted to find out how many are possible, you say, okay, 38 times 5 times 1. Okay, and it gives you some kind of number. Whatever, 40 times 5 is 800? No, 2,000, 2,000. 200? 200. Oh my gosh, yeah, not a good math brain day. So, okay, neighborhood of 200 possible light chains. I'm not going to even attempt to math the rest of these out. But lambda chains, which you could also have instead of a kappa chain, because why not? Why not? Yeah. You got ballpark 30 different variable regions you could select. You got either 4 or 5 join regions and either 4 or 5 constant regions. And so multiply like 33 times 5, and you get your total for light chains. And then any of those light chains can go with any of the heavy chains that you can make. And so the heavy chains, you're going to have, somewhere around 40 different variable regions, 23 diversity regions. That's diversity. Yeah, yeah. 6 joins and 9 possible constant regions. The different constant regions are the different antibody types. So there's a mu chain, which makes it an IgM. We classify antibodies based on which heavy chain constant type they ended up with, because that's the thing that cells are going to be able to grab onto. It's the constant region. Yeah. The light chain has a constant region. It doesn't really work like the constant region of the heavy chain. It's just that there's less diversity there in general. Yeah. And it's analogous, like the way that it's put together. So, all right. So, why not look at that? Let's look at that. So, here we go. These are the way the genes are actually going to be arranged. And so the lambdas are on chromosome 22, the kappas are on chromosome 2, and the heavy chains are going to be on chromosome 14. Do you need to know that? Not particularly, unless you're doing human genetics, in which case it may be interesting. If you have a defect in one of these chromosomes, it could end up affecting that chunk of the immune system. Like if you get some weird recombination or like a fragile chromosome kind of thing. Yeah, it can be bad. Yeah. So that's how they're hooked up. And so in the process, we're going to delete all the ones we don't use. The cell is going to commit. And do you remember? Do you remember which thing it commits to first? The DJ. Yes. Always commit to a DJ, whether it's your wedding, whether it's your immune system, anything in life. Yeah, there you go. That's a good lesson. Good lesson to live by. All right, we're going to jump past some of this stuff because it's there, and I'll show it to you, but this is just how stuff works. There are regions that are going to get joined together, and it's always going to be a short region that's got a 12 base spacer, and it's going to end up joined up to a region with a 23 base spacer. These two things are going to get put together by an enzyme, and we will get to the enzyme because it's coming. And so, yeah, we end up looping all these things out. And so once the DNA for those extra domains that we're not going to use is made, it gets chopped out. It's no longer part of the chromosome. It is extra chromosomal DNA. Yeah. Which will then go on to get degraded / lost from the cell. Just like a little circle of DNA with a bunch of genes that cell's not going to need anymore. It's literally burning bridges. That's just the way the immune system works. Step 1, burn some bridges. Yeah. Step 2, I don't know. Yeah. All right. So, yes, we do all these things, and we have all these enzymes involved, and they're useful. So you want to see how this happens. Fine. Let's see how this happens. So the main enzyme that does the recombination, the VDJC recombinations or VJC recombinations, is called RAG1 and RAG2. And RAG1 and RAG2 look almost identical, and they form a heterodimer. There we go. Because they're different proteins, but they're kind of the same protein, genetically, evolutionarily. Yeah. And so, yeah, RAG1 and RAG2 are going to assemble, and they're going to hold on to the large region, which is 23, and the small region, which is 12. And there's sometimes when the 12 is going to be on this side and the 23 will be on the other side. It takes you through all those in the book. There are lots of possibilities, but for us, as long as we know the path is good, and the path gets us there. So then what you get are, so the RAG 1 and 2 are actually going to chop out everything in between the particular 12 and the particular 23 that they happen to have landed on. Yeah, neat. So they are going to chop it, and then depending on whether it's a bit that is going to code for part of the protein, there are a different set of enzymes that come in. The last step here, I think, is the important one. This is a thing called DNA ligase type 4. And the XRCC is telling you the type of ligase that it is. But DNA ligase 4 is good enough for me. And DNA ligase, so the RAG complex, RAG 1 and RAG 2, those cut the DNA. And then DNA ligase 4 is the thing that's going to re-glue the DNA back together, missing the chunk that RAG cut out. So RAG cuts it out and holds it a little bit away, and then DNA ligase 4 will join it back together. In between, you can have all kinds of diversity added. So there's an enzyme called terminal DNA transferase that can add nucleotides onto the end. And you've got these other things that can actually modify some of the nucleotides. So at the points where you make these junctions, you can actually add nucleotides and amino acids that they code for. You can take away nucleotides. You can, mutate nucleotides effectively. And so because of this, you don't necessarily end up with an antibody that's just a simple combination of, diversity number 7 and variable number 25, pushed right together. You're going to end up with a little bit of chaos at the join site. And that little bit of chaos, gives us, it's a powerful thing, because we can then modify an existing, small set of maybe a few tens of thousands of antibody combinations, and we can turn that into virtually limitless antibody combinations. Probably is infinite technically, just because you could theoretically keep adding nucleotides to that junction until you make this giant, infinitely large antibody molecule variable region. Yeah. So the third hypervariable region is the one that comes from either the diversity or the variable exon, and so that's also going to be the point where we put those together. So that's going to be a point where we have basically somatic cell hypermutation. This is the process where we just end up doing crazy things down there. Here is RAG1 and RAG2. And so they've drawn it so an alpha helix is like a little cylinder. So you can see they're pretty much the same protein. They're just hooked up back to back like that. So they've got a little beta barrel down here, a nice beta stranded top, and a lot of alpha helices that are forming the joining section where they wrap around each other. You can see the green one starts out on this side, goes over on top, and ends up kind of underneath the blue one. So they're twisted around each other, kind of like that. And these regions on the top are able to bind zinc. Zinc has a positive charge. It's a cation. And cations like to bind to negatively charged things, such as the phosphate backbone of DNA. So having the zinc there will help this thing to, attach itself and stay attached to DNA. And then these little purple domains will actually scan along and try and find the 12 and the 23. You know, one of them likes the 12, the other likes the 23. I don't remember which. You can look it up if you want to. I'm sure it's in the book, actually. And you also don't need to know that for this class anyway. But yeah, they look for that particular sequence, which is invariant. And once they find it, they bind down on it. And so, yeah, just imagine RAG sitting here and pulling DNA along until it gets something it likes, and then, ooh, like RAG 2's bound, and so RAG 1 is just going to until it finds something it likes, and then, boom, they're going to come together and cut the DNA, and all that loop of DNA that they cut out then just floats off and goes off and dies. And the enzyme that's going to clean up the mess is going to be DNA ligase number 4. I believe it's XRCC. Yeah, is the motif that makes it work. Okay, so here you go. It's that same thing as a picture. So we're going to take a variable region, and it looks like RAG1 is going to bind the short end, the 12 base pair. Looks like RAG2 is going to bind the long end, which is the 23 base pair repeated sequence. And then, yeah, it's going to, boom, come in and cut right there. Oh, sorry, it's 2 copies of RAG1 and 2 copies of RAG2. Sorry, RAG2 is the top. I forgot that. I forget that every time. So our purple bit back here is RAG2, as it's labeled on the thing. Oh my gosh, yeah. Sorry about that. And these are 2 copies of RAG1. Yeah, so they're going to bind to their little things, and that's amazing. All right, so jumping ahead. I think, yeah, so this is just explaining how the combinatorial diversity works and the basically hypermutation at the junction sites. So one way is we can add on nucleotides, and it's that terminal DNA transferase, T little d T, terminal deoxynucleotide transferase. Yeah. So it will transfer DNA nucleotides at random, completely at random, to the end of a chain. And so if it happens to land on the chain, you better hope that it puts in some multiple of 3 nucleotides, because otherwise you're not going to have a working open reading frame. Make sense? Because remember, nucleotides are read in groups of 3. And so if you put in 2 or 4 or 1 or 5, then what you're going to end up with is you're going to throw the rest of the coding sequence that would make the constant region, you're going to throw that out of frame, and so you're not going to be able to translate it. It'll still be there, but it'll be in a different reading frame, and so it will not be read. Yeah. And so some of the time this works. It's kind of a high risk, potentially high reward play, and some of the times it fails, and that's okay because you have a lot of cells trying a lot of things. This just gives them an extra kind of mechanism to seed a little bit of chaos into the system. Yeah, that's what you want. All right, fine. That's good enough for that section. We'll do one more section, and then that's going to be it for Chapter 5.